So far we've been looking at the experiments where Mendel used data to help create his law of segregation. And he tracked these single characters, and the one we've been using is flower color, with the two different traits, purple flowers or white flowers, with purple being dominant. And the F1 offspring were these hybrids, so he called these monohybrids because they were the hybrids of true breeding parents for a single character. Now, it wasn't just flower color that Mendel tracked. He also looked at six additional characteristics, six additional characters, including seed color, the texture of the seeds, pod color, the shape of the pod, the position of the flowers, and whether the plant was relatively tall or short. Now, on the tests and in working these problems, I'm going to expect that you know a couple of these, like flower color or maybe even seed color. But as far as just sitting and memorizing this entire figure from your notes, don't spend time doing that. You should be able to look at the information that's given and you'll either be told what is dominant, or if you look at the notations of the alleles, you'll be able to tell what is dominant. Recall that when we did our Punnett squares for these monohybrids, the dominant allele was always capitalized and the recessive allele was always lowercase. So in this case, if you see a capital Y and a lowercase y for seed color, well, you know that the yellow is the dominant trait while the green is the recessive. Now, Mendel came up with the second law called the law of independent assortment. And in doing this, he used a dihybrid. In other words, he was going to track two characters at a time. So again, he started with two true breeding pea varieties, in this case, yellow round seeds, and he crossed them with true breeding green wrinkled seeds. Again, from looking at this notation, you can see that yellow and round are dominant, while green and wrinkled are recessive. The F1 offspring are known as dihybrids and would all have the heterozygous genotype for both alleles. Now, when you're working these Punnett squares, remember to group your like alleles together. And if you have a dominant allele, remember that you always place it first to remind you that this is the phenotype that should be expressed. Now, when Mendel performed these dihybrid crosses, he didn't see the typical phenotypical ratio of 3:1 like we saw with the monohybrids. So what this means is that these alleles are segregated or are going to separate at some point. So the alleles will independently assort themselves. The two alleles for the first trait will be paired with one of the alleles from the second trait, and they will sort independently. And we can see that now if we look at this Punnett square. So here is the parent generation, and again, true breeding, dominant allele for round, and two dominant alleles for color, round and yellow. Notice that the other true breeding parent generation, homozygous recessive for both genes, is green and wrinkled. And I like this figure because it also reminds you that these are alleles that are located on chromosomes, and we're talking about homologous pairs of chromosomes. After meiosis, each of these will contribute 1/2 of these homologous chromosome pairs. So our F1 generation then is a dihybrid, possessing a dominant and a recessive allele for each gene. And the phenotype of the F1 generation is round and yellow in this case. Now, when we cross the F1 generation together to produce the F2 generation, well, this is where you'll need to carefully create your Punnett square. And to do this, remember that these alleles are going to separate. And so you always want to pair your alleles together, usually using one parent across the top and one parent across the bottom. Now, This can be a little bit tricky, so you need to take some time and practice these. And there's going to be several different places you'll find practice problems in order to do this. But if we have this individual's genotype as one parent, we're going to take the first allele, a dominant allele, and we're going to pair it with the first allele from the second gene. So 2 dominant alleles here. Then we'll take the first allele and we'll pair it with the second one here, and then the second allele paired with the first one here, and the second allele paired with the second one here. And we'll do the same thing for each parent. Then all you need to do is pair up the alleles. Again, making sure that you put the like alleles together, the alleles for the same character together. And if you have a dominant allele, make sure that it is written in your notation first. Now you'll notice that the ratios are different now. And in fact, our Punnett square has 16 squares to it, 4 possibilities along each side. 4 by 4 is 16. So the ratios are 9/16 with the round and yellow phenotype, 3/16 with the round and green phenotype, 3/16 wrinkled and yellow, and then 1/16 that is homozygous recessive for both genes, and that would be our wrinkled green phenotype.